Now the next speaker is Eva Carro. She's also from Madrid, from the 12 de Octubre, and she's going to talk about metabolic disorders and inflammation as risk factors for Alzheimer's disease. Thank you, Eva.
Okay, thank you. Well, first of all, I would like to thank the organizers for inviting me to participate in this session. No, I'm sorry. Okay. Well, Alzheimer's disease is an aging-related multifactorial disorder characterized by a number of pathological events. But in Alzheimer's disease, some of these characteristics, for instance, glial activation, the inflammation, or synaptic loss, may be accelerated, may be disrupted by many factors, not only by beta amyloid or by neurofibrillary tangles, but also by hormones, lipids, lipidemia, and obesity. Although the exact underlying mechanisms are still unknown, it's a fact that metabolic disorders, including diabetes, obesity, hypertension, and dyslipidemia, have been proposed as risk factors for Alzheimer's disease. Okay. Many of these findings are based on epidemiological data. Many studies have reported that an excess in body weight during middle age is linked to an increased risk for Alzheimer's disease. Clinically, obesity measured as body mass index greater than 30 at 40, 45 years of age is associated with threefold increase in the risk of Alzheimer's disease. It's possible that the association between this increased risk of developing Alzheimer's disease and this obesity, this excess in body weight during middle age, reflects a diet high in simple sugar, fats, and sedentary lifestyle. In fact, recent studies have reported that an adherence to a Mediterranean diet with increased physical activity can be protected against Alzheimer's disease pathogenesis. One of the earliest alterations in Alzheimer's disease is reduced cerebral glucose utilization. As we can see by PET imaging. In fact, this technique is currently used as a biomarker, as an Alzheimer's disease biomarker. There are multiple factors or causes that may be involved in this cerebral glucose hypometabolism. Some of them may be summarized within metabolic syndrome. Metabolic syndrome as a disorder characterized by multiple biochemical and physiological alterations and associated with impaired signaling of insulin and also of other adipokines. In fact, you know that, as we mentioned today, some authors have proposed Alzheimer's disease, sporadic Alzheimer's disease, as a brain diabetes or type 3 diabetes, a form exclusively affecting the brain. Metabolic syndrome is a cluster of abnormalities with metabolic origin, and the central mechanism is closely related to mitochondrial dysfunction. This mitochondrial dysfunction can be induced by accumulation of beta amyloid within these organelles, acting as a direct toxin and beginning a cascade of alterations, including dysregulation of mitochondrial calcium homeostasis and leading to neurodegeneration and even the death of the cell. But these metabolic alterations may be, or they may appear very early, even after the appearance of AD pathogenesis. Well, we will later come back to this point in the next slides. Well, as we mentioned before, Alzheimer's disease share many features, many characteristics of diabetes, including the impairment in insulin and IGF-1 signaling pathway. In Alzheimer's disease, there is an early insulin signaling alteration in insulin resistance, associated also with a resistance in IGF-1, and also accompanied by dysregulation in insulin receptor S1, inducing the cognitive decline. Thus, the IGF-1 resistance, measured as reduced levels of IGF-1 into the brain, is involved in Alzheimer's disease. Here we can see only a couple of examples, a couple of studies where this IGF-1 resistance have been tested. In this first study from the group of Torres Alemán, they tested and confirmed this resistance in IGF-1 both in mouse models of Alzheimer's disease and also in patients with this disorder. And in this other study, we described polymorphism associated with increased risk for Alzheimer's disease, but also with enhanced circulating levels of IGF-1. And this later data was explained, or we suggest that this increased in circulating levels of IGF-1 could be a compensatory response trying to, because, it could be reflects a resistance in the capability to enter this IGF-1 into the brain. Of the metabolic molecule also involved in Alzheimer's disease is leptin. Leptin was previously described as a molecule with multiple functions in the brain. Well, here we can see the effects of leptin in the process of neurogenesis. It was already published in vitro and in vivo models, and we only confirm this capability in transgenic mouse model of Alzheimer's disease using lentivirus lentiviral vector overexpressing leptin. There are also studies, previous studies, where it was reported the effects of leptin over other kind of other processes. For instance, reducing the accumulation of beta amyloid, and also modulating the phosphorylation of tau. And in this other study, we confirmed this capability of leptin to reduce brain accumulation of A-beta, but also we report its effects modulating synaptic plasticity, the length of neurite length, and also modulating the cognitive and behavioral function. I would like to mention other interesting neuroendocrine peptide named aminoprocalcitonin, MPCT. MPCT was previously reported as a suppressor of food intake, modulating the expression of PONC and MPY. And it was also upregulated in processes, in sepsis and inflammatory processes. And we performed a recent study in APP-PS1 mice, where we found that we proposed that brain amyloidosis in these animals, and we also in samples from patients with Alzheimer's disease. Brain amyloidosis was linked to increased brain expression of MPCT, and the potential mechanism could be by the stimulation of pro-inflammatory cytokines. Well, then, in summary, we have many factors, hormones, leptin, insulin, cytokines that are regulating the processing of APP, the trafficking, the APP trafficking, even the clearance of amyloid beta, but also these molecules are modulating other processes, for example, neurogenesis, microglial in general activation, oxidative stress, mitochondrial dysfunction, and all of them leading to neurodegeneration. It's a fact that the concept of Alzheimer's disease is changing in these very recent years. Today, very briefly, in this session, in this presentation, we are talking about the dysregulation in lipid or in glucose metabolism that are affecting the processing of APP, affecting phosphorylation of tau, and inducing neurodegeneration. But in this context, neuroinflammation could be a central player, could be a connection or a link between these processes, metabolic disorders and Alzheimer's disease.
Eva, 5 minutes.
Yeah. Only to mention this very interesting review from Michael and Eka, where they point about several events or several situations that are known to be risk factors for Alzheimer's disease, and they are a common component, and this is a systemic increase in pro-inflammatory mediators. These pro-inflammatory mediators come from newly concepts, newly lexicons. Inflammaging, metainflammation, as an inflammation associated with obesity. Then these inflammatory mediators, systemic inflammatory mediators, can enter into the brain, can cross the blood-brain barrier, and act over the microglial cells, and leaving the CNS, the central inflammation. The activation of these microglial cells involves an increase in the secretion of more inflammatory cytokines and chemokines, and then it it may induce to a vicious circle affecting other surrounding cells, astrocytes, neurons, leading to neurodegeneration and neuron loss. But this microglial activation can be induced not only by beta amyloid, can be induced by other factors independently of beta amyloid. For example, systemic inflammation or activation of the immune system. Then, neuroinflammation could be induced by central intrinsic factors, but also by systemic influences. Let's go to see very briefly a couple of examples. In this study, authors, they activate the immune system in very early stage, in maternal and postnatal stage, in wild-type mice, and activation in the immune system produce alteration in the APP processing, in the tau aggregation, and inflammation, activation of microglial cells. In this other study from our group, we try to explore this hypothesis, and we use this transgenic mice, EMD, deficient in megalin endothelial cells, that they were previously reported as a model of neurodegeneration with cognitive deficits, and more recently, we found that these mice exhibit obesity and metabolic syndrome, and neuroinflammation with an increase in pro-inflammatory cytokines, reduced neurogenesis, and mitochondrial dysregulation. Then we can suggest AD-like pathology.
Eva, you are running out.
Yeah, in summary, many targets, many signaling pathways involve metabolic syndrome and neurodegeneration, not only beta amyloid, and conclusions. We have more and more information, fortunately, more and more pieces of the puzzle, but I'm afraid that the puzzle remains incomplete, or we are not putting the pieces in the correct place. And recommendation, check your lifestyle. And thank you so much for your attention.
Thank you very much.